So in this lab at the moment we're growing algae, and the columns behind me are a kind of bench scale, what we call reactor. In this case it's a photoreactor, so a photobioreactor, so it's essentially a glass tube which is mixed through the injection of gas and surrounded by lights. There's something about this one that's a little bit special. One is that it's very high in an omega-3 fatty acid. The omega-3s that fish accumulate come from the algae in the first place. So it makes sense to look at algae as an alternative source as opposed to fish oil. But in order to do that, we have to boost yields in a sort of algae farming way, and the only way to do that is to use technology. The reason algae like this are a bit harder to grow than, say, a yeast is that you can grow a yeast in a large vat and stir it because it doesn't need light, whereas with the algae we have to provide it with light. So if you have it in a large container, once the cells become dense, they self-shade, so the ones in the middle won't be getting any light, so they won't be growing. So you have to come up with ways to grow them more efficiently than that. We need the input of various types of science. So one is definitely from the engineering field. So the large reactors that we use at this scale are quite sophisticated and expensive, and a lot goes into the design of them. So that's where the engineering comes in. But biologists also have a role because we know how to characterise the growth preferences of strains like this, and for that matter, finding them and identifying them in the first place. You know, that's kind of what we do. So it's a nice collaboration between technologists and biologists.